What we will be speaking about today is the title of the talk is Beyond Public Data, how ultimately proprietary protein ligand complexes can be used to customize co-folding models. And we will look at both fine-tuning workflows, but as well, we will present a case study of a federated version of that model that we built in collaboration with 5 large pharma companies. So we'll share more about the results and the experimental setup of this initiative as well. So to introduce the topic, I think many people and probably many people in this room are excited about the potential of co-folding models. If they would work, they can meaningfully change numerous drug discovery workflows. In particular, bringing in structural information earlier into the drug discovery design process, but as well with the latest generation of co-folding models that as well predict binding affinity based on structure, we can look at use cases such as virtual screening to prioritize hits, or as well lead optimization use cases. Now, many people see the potential of the models and why many people are excited about it. However, when we look at the applicability of these models to date to these various workflows, we see as well very often these models don't work well yet. I'm excited to hear as well the talk that is happening after our talk here, where I think as well Boltz 2 is being used to look at a real use case, and I'm excited to hear kind of the experience there. But generally, what we see with the current versions of co-folding models is that they perform well if the problem that you are throwing at them is well represented in the public data domain, in particular the protein data bank. However, if the problem we are looking at is outside of distribution to the PDB, then very often these models perform poorly. So examples of that are if you're trying to place a ligand in a protein conformation that is not well represented, if the protein forms part of a multimer with, for example, a very large sequence size, or as well, and there have been great research papers on this, if you're trying to place an allosteric ligand in a protein where in the PDB there's mostly orthosteric ligands and those are overrepresented. Then very often co-folding models do not work well and don't fulfill the promise yet. Now, the reason for that, and many performance benchmarks that you see have a similar structure than what you see here. You essentially look at, you cluster different bins according to the similarity to the training data set. So this is looking at similarity to the PDB. And then you're looking at different model versions. And essentially what you see is if the models are very similar to the training data set, they perform well. However, the further you go away from the training data, the poorer these models perform. And that's consistent pretty much across all the respective models that are right now available. And so when you look at that, what many companies or research institutes that work on problems find is that for their specific problems, these co-folding models do not work well. And so the question arises, what can you do in order to improve these models for the given research tasks that you're looking at? And so in particular, the whole topic of fine-tuning and customization workloads are key topics here that we have a lot of experience with and find to very often significantly improve the performance of these models. And that's the main topics that we are speaking about today. Now, what we will show later on is as well how to tap into proprietary pharma data at much larger scale. And so just providing here as well a bit of background in terms of how much proprietary data sits in the private domain. So if you look at the PDB, there are a lot of structures. However, if you exclude all the sugars and metabolites, so anything, any molecules that don't really have drug-like properties, then the number of protein ligand structures in the public data domain is surprisingly small. And so when pharma companies look into their own data repositories, often they have a few thousand structures themselves. And so utilizing those is really meaningful to improve these models. Now, generally, what we find, given that these, let's say, experiences with co-folding models, when applied to your specific research task, what we find is that organizations need the capability to quickly benchmark these models. That's absolutely crucial, given that a priori, it's often not known whether the models work well for your given research task. And so what we generally recommend to start with is, if you have a given research task that you want to look at and apply co-folding methods to start with a benchmarking exercise and look for kind of the problems that you're looking at. Are the predictions that I'm making in line with the ground truth? And if not, you can look at setting up customized models for your specific research problems. And so there are various ways in terms of how you can build customized models, and we'll touch on a few in this talk. But very often, pharma companies look at the following structure. They often start with a public model, or in the case of being part of a network, with a federated model. And then they generate a fine-tuned model leveraging all the data that they have. Now, important here is that as well, public data like the PDB is used as well, in that global fine-tuning run, such that the globally fine-tuned model does not forget what it has previously learned. And then this globally fine-tuned model can be used to make predictions, but as well, very often, you still want to as well generate a program fine-tuned model. For example, in a case where in the global and public database, there are still certain binding modes overrepresented, and your program-specific model should look at a different binding mode. For these cases, making a very specialized model just on a handful of structures can be very meaningful. Now, what we will speak about first, and what Ben will present, is the work that we've been doing as part of the AI structural biology network, where we essentially looked at that fine-tuning paradigm, but took it as well beyond fine-tuning on a single pharma company's data, but actually looked at fine-tuning on the combined data of multiple pharma companies. And so coming back to kind of what I shared earlier, the number of relevant protein ligand structures in the public domain is not that large if you kind of take drug-like properties into account. However, there is a meaningful delta in terms of structures in the proprietary data domain. And so the network that we power here as part of the work that we are doing is called the AI Structural Biology Network, where many large pharma companies are involved. And so in particular on the use case of protein ligand interaction prediction, we did some work where we are happy to share results, and then as well look at how this work continues and what we are doing going forward. So far a bit kind of context to the problem. And so now I'll hand over to Ben, who will run us through the setup and the first initiative that we did here. Ben, over to you.
Hi, thanks, Robin. Yep, I'm Ben Irwin, and I'm principal machine learning engineer at Apheris, and I had the pleasure of working on this initiative last year. So this is the kind of AISB1 run we did with 5 of the farmers within the network. So Takeda, AbbVie, Johnson & Johnson, Astex, and Bristol Myers Squibb, each contributing thousands of proprietary structures that we could train on in a federated manner to train the OpenFold 3 model alongside PDB data as well. So here, data is the real differentiator, and we're testing this hypothesis that a sort of wealth of private data alongside the public data is enhancing the models significantly. Now, this was a big feat to pull off. Executing the training of these models is not easy, and it's something that we've developed at Apheris to do. So what we did in this project, there were sort of three steps. One was to take the test sets for each of the private pharma companies and evaluate the performance of the baseline OpenFold 3 model on them to characterize how well that model was doing out of the box. For each of the companies, we ran an independent local sort of global fine-tuning. I say global fine-tuning for that entire company's data. So that would be a significantly improved model on just their own data. And of course, then a single federated model across all of the companies. And that's exactly what we can see in the summary bar chart here of this performance. So we've got two bars per model type, the protein ligand interaction, LDDT, which is the local kind of quality, and the protein-protein interaction, which is slightly higher. And here what we're doing is we're looking at the fraction of the test set that has an LDDT greater than 0.8. This is a cutoff that's deemed as sort of very good prediction quality, something you could use to do real project work with. And you can see the OpenFold 3 baseline model is getting LDDTs about 30, a bit over a third of the test set is getting a score that is this sort of very good cutoff. The individually fine-tuned models on each pharma does improve significantly beyond that, but the federated model is sort of blowing it out the water and is sort of, over a half for protein interaction, 60% of the predictions are that very good quality. So we're seeing the federated model is superior according to these metrics. And we saw a similar prediction quality in all of the metrics. These are just the most interesting ones to summarize. Next slide. So something you can do to inspect sort of why are these federated models working better than the out-of-the-box OpenFold 3 model. And you could plot, for example, across a data set, the federated models metrics versus OpenFolds. And here you can see a number of cases on public data. We can look at public data. That's all we've got access to that are much better here. And these cases kind of give you a hint at what the pharmaceutical companies might see as improved performance on their side when they look at their own data. So on the right here, one example that came up, a sort of ternary, you've got three kind of non-protein entities within the binding pocket. There's two ions and a small molecule. And the original OpenFold was sort of just couldn't get the pose right, but the federated version had learnt how to handle that accordingly. The next example, if there is one, yeah, this one's a heme sort of group there, but it was upside down. The ligand had inverted, and then the federated model was getting that correctly. So this could indicate then other drug molecules getting their pose corrected in private data sets. Finally, an example where neither the ground truth nor the OpenFold 3 or the federated model agree, but the metrics have improved significantly. So it's a step towards, a step in the right direction from the model. So this is very interesting, and we'd expect each pharma company can see sensitive things on their side that they're excited about in a similar vein. The more exciting plot that sort of gives the whole breakdown of this project, here is a matrix of each model, the top row being the baseline OpenFold, the next row is the federated model, and then each of the 5 rows are the individual models that were locally fine-tuned on the pharma data. And then the columns here are those test sets from the farmers. And it's been normalized per column, and it's been normalized as well. So OpenFold is sort of 0. This is the delta. And you can see that the federated model is just better at every data set across all farmers, whatever. And the individual farmer sort of fine-tuned models are good at their own respective data sets down this diagonal. But if you were one of the members of this consortium and you were choosing which model to use, you would choose the federated model because it's just got such a wide domain of applicability and robust across the board performance. Where are we going with this? Well, as Robin showed, there's binding affinity prediction is on the roadmap for this year. And we've realized that these companies also have a huge quantity of proprietary binding data that can be very coarse data and it can be very precise data. And we've got the opportunity to play with binding affinity heads that are coming out. The OpenFold model is compatible with the Sandbox AQ affinity head, for example. but there's a lot more sort of that can be done in the affinity head architecture space. And the most important thing that I think a lot of the sort of models in the co-folding space are benefiting for is self-distillation, the ability to build up a large sort of set of distillation data that can be used on training to refine the model and grow its performance in a direction. So we can build a proprietary self-distillation set for each of the farmers and train on all of that data as well. So there may be another 10x of data that could be generated using the affinity prediction and the affinity data as a kind of filtering and harmonization type process to generate that data set. What does this mean for the kind of pipeline that Robin showed? Well, of course, now we're going to have lots of different model weights to work with. We're going to have structural weights from things like ASB1, but there could also be affinity head weights. And then if you've received this large federated model, you're going to want to do downstream fine tunings of that on each of your individual newest, latest drug programs. So handling all of these weights live in a project with sort of updates and possibly distillation sets as well is not an easy thing to do. And we really want to empower users to do this so they're not sort of weighed down by the technical requirements there. So what we've done is we've built the Apheris fold software. This software can support the benchmarking of various types of models, OpenFold, Boltz, soon Proteinics V1, which is very good in the large molecule space. You could benchmark that on proprietary in-house data. This is deployed completely within your firewall, so there's no information leakage. It's very secure, and you can securely fine-tune models through the application on data in your own company and operate with either a graphical user interface if you're a medicinal chemist and that's easy to use, or a programmatic workflow. So this is sort of really empowering people, and we're really excited to offer this. If there's time, we could look at a fine-tuning case study, but they might want time for questions now.
Thank you, Ben. So maybe pausing here for questions. Not sure how much time we in total have. Otherwise, we are happy to as well share some local fine-tuning experiments as well.
I think a couple of minutes for questions, and then we need to move on to the next speaker.
That sounds great.
I guess one question from my side. A lot of models, especially in the industry domain, there'll be edge cases, things like bridging water molecules within the protein pocket and things like that, which off the top of my head, I don't believe OpenFold 3 accounts for yet. So are you sort of working those things into your fine-tuning process, or any ways of addressing that? Because that can easily account for outliers, and if you're otherwise incorporating them non-mechanistically, it can basically just tend towards overfitting in the private domain.
Yep, that's a really great question. I mean, there's a lot of considerations in the sort of workflows and data processing pipelines. So firstly, the data processing, we've got validation and preparation scripts that sort of cover getting a farmer's data from sort of raw data into the modeling kind of capacity. But of course, and we're in discussions with OMSF about this as well, because it's a very common concern. On top of a co-folding output, there's also solvation, and there can be fixing of charges and things for downstream computational chemistry and modeling. So it's nice to have all of those voices in the ecosystem informing our kind of data processing pipeline. And yes, we're building sort of people's concerns into the application. In terms of water molecules specifically, I believe you could put those into the co-folding, but it's definitely a big topic in the space. Yeah.
Thank you, Ben. Hi, Mike, by the way.
Hey, how's it going?
There's a question in the channel that I'm happy to quickly cover. So the question is, if the federated OpenFold 3 model will be eventually released, or the weights will be released for public or academic use, or whether that model will remain exclusive to the partners. First of all, this model, as rightfully stated here, is right now exclusive to the partners that have been participating in this initiative. Generally, the ambition is as well to invite further pharma partners that can contribute data to such an initiative to the consortium, which is why as well there is a commercial incentive for them to only allow them to use this model when they join the network. Now the discussion on releasing weights to the public domain for academic use is certainly one that is as well being being discussed within the network. Apheris doesn't own that model ourselves, so it's ultimately ownership of the participating pharma companies, and they decide kind of what to do with it. But of course, providing academic access to that would be very valuable for research purposes. That's the current sense.
We have a question in the room.
Hi. So question in the room, sorry. So Jeffrey here from Bolts. Interested in your federated model. So you've trained it or fine-tuned it on all this additional structural data. Have you done any experiments to show that the model's actually getting better at generalizing, or have you just enriched the data set? And how does the improved performance compare to the additional, what's the similarity to that additional training data?
Yeah. So Ben, do you want to start, or?
Yeah, I can say like we did do some experiments along those lines, especially measuring against the PDB validation set that was sort of held out from the training of this. And definitely across the board, the primary metrics stay at least the same. And then for certain classes like that are really well expressed in the pharma data, like protein ligand interactions and even protein-protein interactions, there is an improvement. Whereas you do see a degradation in things like RNA and DNA, which we explicitly did not include. So there can be variation in metrics, according to fine-tuning.
I think we're going to have to move on. We have 3 speakers. So thanks very much for the really great talk getting us started. And please join me in thanking them.
Thanks, everyone.
Yeah, so thanks everyone for coming. So I'm going to share you some of my Bolts co-folding experience. In fact, this is much more of a lecture than the previous talk. So it's a condensation of a lecture I gave earlier this year for the chemistry department. And it's definitely all about things you can try at home. So just an introduction to myself and where I work. So I work for the Alzheimer Drug Discovery Institute in Cambridge. So that's there on the top left. We're on the biomedical campus, and we're part of the University of Cambridge, and we're part of the Alzheimer's Research UK Drug Discovery Alliance. So we have sister institutes in Oxford and at UCL. In fact, my colleague Anish from UCL, he's also here in the audience. And what we try and do is take the latest science into dementia, and including Huntington's and Parkinson's, and turn that into new drug discovery projects. So from academic collaborators towards validating targets, we do in-house drug designs, synthesis, screening, and then hopefully come out with a lead compound to partner. I am the only computational chemist in our group, so I do all sorts of different things, whatever it takes for a project to move forward. But recently I've been really getting involved with protein folding because it's been really helpful into moving our projects. So here we go. Today I'm going to talk to you a little bit about what came before protein folding, just for the youngsters in the room. And then we'll have a look at how we know that we're getting better. Then we'll have a look at some different protein folding and co-folding tools and how to assess models and what's available, how you can use them, some advanced features. And then we'll end with some of the things that can go wrong with protein folding tools. So hopefully with all of that together, you'll be sort of ready to go if you're not as yet. So a little bit of history on protein modeling. This is not a new thing. It's been around for a long time. The first program modeler came out in the early 1990s on the basis of BLAST sequence alignments and the PDB structures. So we've been doing some sort of protein modeling for nearly 30 years, but it's moved massively from there. as you can see, to AlphaFold to a couple of years ago, and this nice picture from that I've borrowed from a presentation was just before AlphaFold 3. So homology modeling, what is it? Again, just for the youngsters in the room who haven't been around like me since the 1990s doing this work, you find the most similar sequence to the sequence you're interested in that has a crystal structure associated, and you basically line them up. You align the sequences with BLAST, and then you can map your query sequence onto the protein. You can try and fill in some gaps with some force field modeling, and then refine your model. Check that it doesn't have any close contacts using a Ramachandran plot, perhaps, and there you are, a model based on a single template. So it's still there, it sort of works, but you know, we've moved on from there now to some fancier methods. So here's where we are now. We now do deep neural networks, equivariant transformers. We've even moved really beyond the folding simulations into all of these new computer-based methods, but they're all based on multiple sequence alignments. So this is a key term in this whole protein folding. You need to align lots and lots of protein sequences, and that will then derive sort of structural features from similar proteins and just from similar domains. So in comparison with the homology model, you only use one or maybe two sequences. This uses many more. It also means it takes quite a lot longer. This is quite a big job to actually do the multiple sequence alignments. If you want to speed it up, you could also use a protein language model, such as ES7Fold, and then you can bypass the whole sequence alignments. Overall, the sequence alignment based ones are still slightly better. This is AlphaFold against ES7Fold. It does perform slightly better, but protein language models also coming along very nicely. So how do we know what's better than something else? So that's mainly done in these CASP competitions. So these are competitions, and they've been going for quite a while. They happen every 2 years. I think the last one that I've seen a publication on was from 2022, but there's been one since that I think will shortly be coming out. And there they take unpublished crystal structures and complexes as well, protein-protein interactions and protein ligands, and people try and predict these, and results get measured against the ground truth. And you can see from the left-hand picture that year on year, the performance has really improved. The percentage of targets that they've managed to predict within sort of 2 angstrom is now at about 80% for the latest CASP results. And again, on the right-hand side, you can see it in a sort of a graph form with a slightly different measurement, the GDT score for backbone modeling. Again, you see the lines here for AlphaFold. It's now massively improving over previous years, particularly if you tweak some of the features away from default settings. So that's really good news. Now, if you want to get involved yourself, what can you do? Where can you go? If you just want a single protein model, you don't actually really have to do anything. You can just go online to Uniprots or to the AlphaFold structure database and download it. Most of the protein sequences have been sort of modeled into structures, and you will get a nice picture as one shown over here that shows you the model with the confidence in the particular regions. So where it's dark blue, we're quite confident that the residues are in the right place. And then where it's orange, really the system didn't have a clue. So this is always something really important to check. And again, this is all about the C alpha atom, so it doesn't really say anything about the side chain. So it could still be that the actual arrangement in the binding pocket is quite a way off. Now, if you go to the AlphaFold database at the EBI, you get a PEA, predicted alignment error plots, as well as the confidence map. And that allows you to do some other assessments. What that really allows you to do is check whether the different domains of your protein have confident interactions or whether the system is not so sure. So for example, I have highlighted here this little square over here. And then that highlights this helix here. That's quite a long way off in sequence from the main body of the protein. But when we sort of travel up from here to this area, you can see this is still dark green. So this is still expected to have a low error in the positioning. So there's quite a high confidence that this little helix is associated with the core. So that just helps you assess also when you do multimers, how likely the interaction is between different domains. So again, really important to check depending on your protein. If you actually want to build a model yourself rather than download one, there are a number of tools available now, and this is by no means exhaustive. So I do recommend, if you want to find out more, to sort of go to this reference paper. But you know, the starter point is always, of course, AlphaFold. Everybody knows about it, but it's not that accessible. Installation is for academics only, and it's quite hard. It's quite specific in the requirements for the GPU. And the AlphaFold server, again, you can access as an academic, but it's very limited in the ligands you're able to use with it. So myself, I'm a big fan of Boltz 2. You can download that from their GitHub webpage. It was developed by people at MIT. Easy to install, and you can also use it via a collab and various companies who do this sort of fee for service. So Neurosnap, Modal, Tamarin Bio, to name a few. They are actually companies that have big servers and they've installed the software, and you can just go there and submit your sequence, pay your money, and you get your model. And for academics, they even do a few runs for free. So check it out. So what comes out of these folding methods, and a big shout out on this slide for my colleague, Dr. Sami Shaban, who's at the LMB, who allowed me to borrow some of his slides. He has an excellent lecture on YouTube on AlphaFold. So outputs, obviously structures, not generally aligned as CIF files. You can read them into most visualization tools. Then you get this JSON file with the confidence scores. And then finally, you get these NPZ files that are your predicted aligned error. We look in a bit more detail at the Boltz outputs. This is sort of the basic file structure. Under your main directory, you get your predictions directory, and in that you should find a CIF file, a JSON file for your confidence, another one, if you ask for it, that tells you the affinity, and then your NPZ files for the domain interaction. If you do your own run to visualize the per residue confidence, I really recommend Chimera X. That has a really nice interface for just coloring up the per residue confidence, and it can also visualize these PAE, pairwise confidence plots. If we look a little bit more into detail at this confidence JSON file, you get quite a lot of different values here. Obviously, the top one is the most of interest. This complex PLDDT I often use as well. And if you have a complex, particularly protein-protein complex, you might also want to look at these ones with the I, which refers to the interface score. If you do things in a more sort of high throughput way and you get lots of models, it becomes quite complicated to sort of get all of the data out. I'm not a big programmer, so I love to use these pipelining tools. So I use NIME. You can download it for free from their website. That can just read in all of the JSON files from a directory, and you can just make a workflow where you pull out whichever confidence you want and have it nicely organized in an Excel. Again, if this is something you're interested in, I'm happy to share my workflow. Just contact me. So we've had a little look at what comes out, what goes into a protein folding prediction, really just a sequence. So in the simplest form, you just put the sequence in a sort of FASTA format. So this, the A pipe protein is the only bit that you require, and then your sequence. Optionally, you can add a ligand, and that sort of goes in the sort of B pipe ligand then. But if you want to take it a bit further and do some fancier things, you will need to go with a YAML file. So this is the structure of a YAML file over here. Again, you got your sequence, but now you can add things like your multiple sequence alignment. So you can recycle your alignment once you've done one run. If you want to just change the ligands, you could stick with your first sequence alignment, save a bit of time, and yeah, point to that earlier file. I've sort of given you more or less the path there to pick up. And then you can also add this properties tag. So that's what you need if you want affinity calculated in bolts. Okay. And then other things you can do, a bit fancier, are templates, pocket residues, and contacting residues. So for these more advanced inputs, first of all, because I struggled with this a little bit when I first had a go, I've just highlighted what you need to do if you have a dimer. If you have two of exactly the same chain, you need to sort of put them in brackets A, B, and then you can do a dimer folding. And then if you want to add constraints, you need to start counting residues. So if you chop bits off your chain, to take that into account when you give the number. And then you can do pocket constraints. So that's between one binder, one ligand, and they can have multiple contacts with a chain. Or you can do contact constraints, and then they are between two residues, and you need to specify a distance. And then finally, the templates. I had quite a struggle with this. It's quite hard, I found, to get the templates going. In the end, what worked for me was to generate my templates as CIF files with Chimera X. These other tools didn't work for me, apart from PDB downloads. But if you have internal files, Chimera seems to be the way forward. And then the other thing I found is to use this max MSA sequence 32. So that restricts the number of MSAs, the sequence alignment, to 32. Why 32? I don't know. It seems to be the magic number. I looked on the Slack site. That's what somebody suggested. I tried it, it worked. I tried different numbers, it didn't work. What can I say? Then we have the parameters that you put on the command line. So we've already seen, yeah, MSAs either in a YAML file or use the server. Diffusion samples is typically set to 1, but I would really recommend running more models than 1. I usually go for 3 or 5. I mean, AlphaFold's default, I think, goes to 25, so up to you, but then it takes a lot longer. Definitely more than 1. Use potentials definitely makes better physical models. The less clashes, they look better. And step scale, you get more different models if you lower the step scale closer to 1. So that can be useful if you want a few different options. We've seen this one, the max MSA sequence. And yeah, you can specify an output directory. What do you need if you want to do this at home? Definitely a GPU, I would say. You can run it on a CPU with bolts, but it's very, very slow. GPU, the more VRAM, the better. This is my laptop. It sort of works with a medium-sized protein, but it takes quite a long time. And in contrast, on my workstation, and that's not a massive GPU chip, that only takes 5 minutes. And again, to put it in contrast, if I do it on the HPC here, it's 80 gigabytes. I can do this dimer type folding in 12 minutes rather than hours and hours on my desktop GPU. The longer the sequence, the longer it takes, and at some point it will just fail if it's too long for your GPU. And the more solutions you request, also the longer it takes. So coming back to the number of models you need, so there's a nice paper here where somebody looked into this, and that just shows you that the top ranked model, which is the line in black for a similarity to the training sets, and then success rate as the Ligand RMSD compared with ground truth. The top model is not typically as good as the best model of 5 different scored ones, and that's true for both AlphaFold and Boltz. So definitely worthwhile inspecting multiple models. And personally, I find with Boltz, sometimes if you have sort of low confidence regions, they fold into the protein in awkward ways that block your binding site. So sometimes you need more models than one, so you have one that's open. Or you can try and chop off some low confidence areas, but that can have its own problems. That takes us on to limitations of folding methods. We've already heard some of those from the previous talk. I think Ben and Robin did a great job at highlighting sort of the similarity issue. So reduced performance on structures not represented in the training sets. There are other things like conformational changes in a protein. If it has different conformers, the one that's best represented generally tends to come out. There's often a bias towards the most represented activation state, so that can be a problem if you want antagonists when most models are agonists in the PDB. And it's also quite tricky to do affinity ranking based on these models. We've really already seen this with the previous presentation, but I'll just reiterate there is definitely a thing where, you know, low similarity to the training set is a much lower success rate, both in protein ligand complexes and protein-protein. And another thing that I find, and I was quite surprised by, is that also domain choice can have a big impact. So in this example, I first of all modeled just one domain of the sting protein. And I find I struggled to get an antagonist model. So in this model, you see the ligand is more or less in the right place, but my Boltz model in blue does not superimpose very nicely on the ground truth model in magenta and pink, as one of the lobes is not in the right place. However, when I then thought, well, maybe it will go better if I just use the whole sequence, including the transmembrane domain, lo and behold, my ligand ended up in the transmembrane domain rather than in the CDN domain. So yeah, different domains, it can affect how your protein model comes out. Sometimes it's good to chop bits off, sometimes not so good. You have to experiment a bit. I thought I'd throw in one slide to go back to homology modeling. Is there still a role for it? I find it sometimes quite nice to make at least one homology model just to compare with my folded models and see if I'm thinking along the right tracks. Because at least for my homology model, I know which template it used. So that allows you to check, for example, the activation state, whether you have your antagonist model or your agonist model. And also, you know, if you want an allosteric model, sometimes it's easier to do that with homology than protein folding. And finally, you know, you can, with your own homology modeling, sort of more easily add things like water molecules to sort of check what the effect might be. And that brings me to the end. That's just a slide here with some resources. So again, shout out to Sami Shaban, who helped me with some of this presentation. There are also resources at the EBI that have an excellent sort of training slides course you can do online. And of course, Chimera, some tools, some of these fee-for-service people. And with that, I'll conclude my talk. And thanks so much for your attention.
Thanks, Henriette. So yeah, it's Jeffrey from Bolts again. Thank you for highlighting the usage on the open access models. It's not really a question, but just to highlight, the Bolts Lab platform allows you to do a lot of what you've done, what you're showing, and those frustrations that you're having. We're actually doing an online demo tomorrow at 9 a.m. So if you go on our website or on our LinkedIn, you'll be able to sign up for that. And I can give out free credits for anyone who wants to have a go with the Bolts Lab platform. Just find me after the talks today.
Good stuff.
Very good. Making links here. There are online questions as well. Aishwarya, do you want to read your questions so we have some dialogue here? Otherwise, I can read that as well, as you feel comfortable.
Yeah, I can read. I just didn't want to interrupt, but I wanted to ask with respect to the you said you have a decrease in success with decreasing similarity to your training data. So are you currently working on improving generalizability?
Not me personally. So what I do for my project is use templates where I have my own sort of crystal structures, which we have for some projects, and that really helps with the predictions, but not so much generalizability. That's just for specific cases.
Yeah, I see. Yeah, thank you for the talk, by the way. Super interesting. And I'm really interested in Parkinson's disease. So it was nice to see Alzheimer's, and a shout out to that too. Yeah, thank you.
We have one in the room.
Yeah, thank you. I'm sure many people learned a lot. There's another question online. Actually, I'm very glad to say that Chris Sander is here as well. Chris, please go ahead.
Okay, hi, it's Chris Sander here. We did EV-fold in 2011 for the first time from covariation folding proteins. But the question I have is that, to Henriette, do you have any information about the coverage of the human genome? Lots of flexible regions, disordered regions, some people call them intrinsically disordered, whatever. They're flexible, hard to predict in terms of structure. What fraction do you know of the human genome can be covered by any of those methods? I think it's close to 40%, but do you have the actual number?
No, I have no idea, but it's a really interesting question. I don't know whether anybody else in the room has any ideas. No, I think we can't answer that question, I'm afraid.
Yeah, sorry, really nice talk. Thank you. I wanted to pick up on something you said in the beginning. You said that it's helped you a lot. So I was wondering, could you how has it helped you and compared to what you were doing before?
It's helped me a lot, I think, in doing projects, because we have quite a few projects where there is really very low similarity to what's known out there. And it just helps to get more hypotheses. So we still don't know whether those hypotheses are correct. But then we can use the models to do docking and see whether it starts to correlate with the data that we generate. And that then helps, again, sort of generate more of the right compounds. Yeah, so that's how we use it. So we don't know whether our models are good, but we can see from, the increased potency of the ligands that we're thinking along the right lines, and it explains something.
Thank you.
I think we should probably move on there. Thank you very much, Henriette. Our final speaker today is Nicolas from the EBI, who's going to be talking about SureChEMBL with added disease and protein annotations.
Yeah, I guess. Thank you. Hello, everyone. It's a pleasure to present here today. I was quite interested to see that I was invited to speak as part of a co-folding special, even if I'm not going to talk about co-folding at all. So thank you if you're staying for my talk. Indeed, I'm going to talk about SureChEMBL, which is our database of annotated patterns. And in SureChEMBL 2.0, we introduce disease and protein annotations. But first announcement, there will be a second announcement at the end. The first one is that the chemical biology services team at DBI is organizing a UGM in June. So that the perfect opportunity if you're a user of our services and you want to present what you are doing, or if you want to hear what we are going to do next, if you want to influence the direction we are taking. So it's, yeah, you've got the link. So hopefully we will see plenty of you there. So why am I talking about patents? And so brief introduction, why searching chemical patents is useful. And well, it's written, but you want to make sure when you start working on a new compound that you have the freedom to operate. That means that your invention is not already covered by another company. Obviously, also interesting to see what others are doing. You don't want to repeat what is already done, but also strategic to see what others are doing. A lot of science is described in patents, even if it's not always well explained, and also citations and references are you've got everything that is mentioned in the patents. And so obviously there is some unique scientific knowledge there, and that's not only for compounds, but also for compound and protein relationships. And it has been demonstrated that often the information about a compound target is mentioned in a patent way before it's published in the traditional literature. But it's very useful, but unfortunately, it's very difficult to work with patent data. They are freely accessible, yes, typically in a PDF or in XML. That is the result of OCR extraction of the text and the images, and you get an XML. And also, it's not annotated. So very challenging documents. They are also very long. It's frequent that a patent is more than 100 pages. So it's, if you have to read everything, no, you don't do that. The vocabulary that is used, yeah, they talk about science, but yeah, it's not scientists who are writing patents, or not entirely. So it's tricky to read. Obviously, information is quite obfuscated. I mean, yes, a patent protects an invention, but it's even better if no one knows exactly what you are going to do. So it's not described in a way that you know exactly, oh, this is this compound and this is for this therapeutic area and the mode of action is this. No. Information is hidden or is related to the obfuscation of the data, is not provided in a way that it is easy to access. Compound structures, except for the US authorities where you have access to a MOLFI or CDX5, compound structures are in images, same for tables, but a table as an image. And obviously, not only one authority, many of them, and they all have different ways of representing the data. And of course, even worse, you've got 6 million patents published last year. So to imagine doing a manual annotation, it's completely impossible. So with this matter of fact, this is where SureChEMBL arrives. So in very briefly here, it's a database that provide annotated patent. The annotation is done automatically. It was first developed as a commercial product by Digital Science, but in 2013, the EBI was given this service and rebranded it SureChEMBL, and since that is maintaining and developing it. Last year, we introduced SureChEMBL 2.0 because we thought we reached the next milestone with a lot of new improvements. And also, we believe that, I mean, despite the dozens of tools that provide annotated patents that you can find online, most of them, the large majority of them, are commercial products, and they cost a lot. As far as we know, we are one of the last one remaining. The latest news we had on Google patents were not very good. So, I mean, this is unfortunately the situation where we are at the moment. We offer more than 100 million patent texts through the user interface. Obviously, we don't annotate all of that because we are mainly focused on chemical-related patents. But we extract 31 million compounds from 43 million patents. And more recently, we started annotated the text with biomedical entities, and from that we extracted 1 million of these new entity types. What do we do with SureChEMBL? So we aim to have a coverage of the patent which is as large as possible, but obviously it is limited because getting access to these patents demands financial resources. But at the moment, we can offer patents from the USPTO, EPO, WIPO, also the Chinese and the Japanese authority, but with less information compared to the other three. We automatically annotate the full text when we have access to it with chemical and biomedical entities. And there is no manual curation behind. We also extract compound structures from images. We allow through our user interface several types of search, text search with a keyword-based approach, and also structure search using exact structure, substructure, and similarity search. Where SureChEMBL is also unique is that we offer the annotations for free to everyone, and with a very generous license, so anyone can download the data and use them as they wish. And we release a new version of our full annotations every 2 weeks. For the compound identification, as I've already briefly mentioned, we get the information from for the different authorities we have access to, and then from the text we extract the compounds directly. We also extract the structures using image to structure conversion, and for the US patent we get the structures from the MOL files. All the compounds are registered in the same way. We use RDKit and the Campbell standardizer to standardize the molecule, register them, and calculate some properties, molecular properties. Just to show you how quickly the quantity of compounds evolves over the year. It shows from 2015 to 2025 how much more patents we got. Obviously, there is something to mention here that, yeah, we started including Chinese patents between 2024 and 2025, but you can see that it increased dramatically the number of unique patents, but not the number of compounds. And that's because we don't have access to the patent images for the Chinese patent. So it shows that a lot of chemical information is mentioned in the attachment of the patent, and unfortunately, at this date, we don't have access to everything. We're working on that, but it's challenging, as you can imagine. So SureChEMBL 2.0, the recent development that I want to mention here is that we now annotate the patent using LeadMine developed by NextMove. We also started extracting the structures from images using MolScribe, and we now deliver the data through a unique system, which we call the bulk data. And as mentioned previously, it's a full annotated data that we extract from the patent. And also we have an API, which I forgot to mention here, but that you can use to access this information, except the biomedical annotation. They are not in the API yet. So why do we use LeadMine for our annotation? Before, and when I mean before, it was until early last year, we had, I mean, historically, we were annotating only chemical entities. That was SureChem, and then SureChEMBL was doing. And we were using multiple tools to extract the company information from names, IUPAC names, miles, INCHI. I mentioned here a few tools that we were using, but we had issues, I mean, with the price, yes, but also with the licensing. And every time we need to renew the contract, there were long discussions, and it was not ideal. For the biomedical annotations, we had developed our in-house NLP model to annotate gene, proteins, diseases, and mechanisms of action. But the annotation was good, but we had no normalization of the entities. And at the end, what you want is not only to highlight few chunks of text, you also want to know to map these entities to ontologies or control vocabularies. And that was in a state that we were not very happy with that. Things happened, and at some point, we decided that, yeah, we could use LeadMine that does both annotation, biomedical and chemical, does the normalization, and for us came at a lower cost than what we were paying before, and with a favorable license. So this is a sample of the information you get with LeadMine for the biomedical annotations. And that's what you get when you access the data, the bulk data. You can download them through our FTP. And also happy to say that we recently published a new version of the biomedical annotations. And you now have access to the 2025 data. Yeah, so as I mentioned earlier, there is over 1 million biomedical entities extracted from the patent. You can see that most of them come for the type of gene or proteins, but also huge chunk from diseases and less for mechanism. The mechanism entities that provide a large, broad type of annotations is not normalized, or it doesn't have a resolved form. But you can see that when the annotation is found in an ontology or a dictionary, like for this genes or these diseases, there is the mesh term, for instance, the mesh ID, or the UniProt, or human gene ontology ID, where it comes from. The biomedical annotations are not yet visible in our user interface, but we are working on that, obviously. And also we provide the raw data that we get from LibMine. There is no post-processing or filtering, so that's on the user, in the user ends to do that. A bit more on the image compound extraction. We have been improving that by changing the tool that we were using to extract the compound structures from images. For that, we benchmarked several of them, comparing our existing approach in green with the two tools that we selected for this benchmark, MolScribe and Decimer. And here what I highlight is that MolScribe in red is able to find a much more exact structure. So that's, I say exact structure because in this benchmark we could compare the extracted image, the extracted structure from the image to the compound in the MOL file. That's only for USPTO patents, obviously. Yes, so after this result, we decided to implement MolScribe in our pipeline, and that's still an ongoing task, but we are soon finishing it. Having all this new annotations is great, but obviously, if they are not accessible to our user base, it's not ideal. So we used to have several downloads for several different kind of usages, but it was very hard to maintain. Instead, we decided to use only now the bulk data. So that's a collection of core files that represent the entire annotated SureChEMBL data. We are making use of Parquet file because they are highly compressed, but also easy to query. So you can download the files and do efficiently your own large-scale analysis. In this Parquet file, we are not providing the full text. I know it comes as a disappointment for a lot of users, but we provide the patent metadata, chemical and biomedical annotations, and also the locations where this annotation are found in the text. By location, it means the title, abstract, claims, or description, not the exact position in the text. So having this all this annotation now in SureChEMBL, it's very interesting. And what can you do with that? Obviously, as I mentioned before, there is a lot of science mentioned in patents, and so I wanted to show a few examples. They are relatively simple to implement, and that's why I picked them. But you can answer much more question, I'm sure. So let's see. If you want to know when was imatinib first reported, so sorry for the SQL-like code here, but it just shows that in a few lines of code you can query the files, and it gives you this patent, which is likely to be the first one that mentioned imatinib, given the date and also the assignee, which was a Swiss company that was working with Novartis at this time. You can also find easily how many patents are mentioning imatinib with a protein ABL, so it can be ABL1 or ABL2 or BCR ABL, as long as it was identified as containing ABL. And you can see almost 22,000 patents with this combination of drug, this compound and this protein. But even more interestingly, that you can use the location of this annotations where it is, because it matters where an entity is mentioned. If it's in the title, it's even more important that if it's in the description. So here I'm looking for imatinib in the claims and the protein ABL in the title. And of course, it returns much less patents, but they are maybe more interesting if it's what you are after. And here, let's say you want to know which were the diseases first mentioned in 2025. It's quite interesting to see that there are not many. It's not the top 5. It's just that based on the annotations we got, I could only find this 5 diseases mentioned for the first time last year. And we can do a similar exercise for the proteins. This time it is the top 5 because there are much more proteins that were introduced for the first time last year. But interesting to see that this 5 here are apparently they were never mentioned before, and some people started mentioning them only last year. To finish, I want to mention here that we already extract the compounds, and we extract also the proteins, but what we don't have at the moment is the bioactivities. And earlier this month, we have a postdoc who started to work on that as part of an Arise to fellowship in collaboration with a pharma company, and she will be working on extracting the bioactivities from patents and ultimately loading this information directly into SureChEMBL. So we are quite excited with this project. I hope it delivers well. Right. So to conclude, with SureChEMBL 2.0, we have an improved infrastructure compared to our to the system we inherited a long time ago, more than 10 years ago. We updated our annotated workflow. It now the coverage is much broader now. Also, it is faster, and we have more control on it. We use the tools that we benchmarked, and we are confident of the performance. We simplified the download process, offering a simple access point with a biweekly full release, offering the user a way to easily download this information and assess the performance of the annotations. There are many next steps, but I wanted to mention again that we are going to start extracting bioactivities from the patent text, and also the UI, which I barely show on during this presentation, is getting more resources at the moment, and we hope to improve it very quickly. I'd like to thank our team working on SureChEMBL, but also more broadly in the chemical biology services, our founders, and my second announcement that I will be leaving the EBI in a few months, and so if you feel free to reach if you are interested. And with that, thank you very much for your attention.
Will answer questions?
Yeah. What time?
Perhaps I can start off. For the biology extraction, will you be going back to do all the historical patents as well, or just new ones?
No, we annotated every patent from the very beginning. I mean, at least I think first one 1888, something like that. But we only show at the moment, because it was simpler to do that, we only provide the biomedical annotations for the patents that also have chemical annotations.
Okay.
Can I ask a question, Chris?
Yeah, sure. Chris.
Oh, hi, hope I'm live. Yeah, very nice to hear progress in SureChEMBL. Yeah, a few questions. Let me start off with the three key ones. One, everyone's getting a bit vexed about the paucity of new targets. Are you getting new targets coming through your annotations? And are these the primary targets rather than secondary cross-screening? Second question was, as we all know, the claiming diseases in patents has more or less catastrophically low specificity. So I don't know how you address that unless you cross-reference with the SureChEMBL paper where they probably describe it more concisely. So yeah, they're the questions. And so yeah, I've just published a book chapter on the manual extraction of SAR from patents, including from the Binding DB folk who've been annotating bioactivity from USPTOs for some years now, and we published that last year. Okay, thanks. Excellent talk.
Thank you, Chris, for that, and thank you for your questions. So the first one, well, we are using LeadMine for the annotations, and of course we rely on the dictionaries used by LeadMine to be as up to date as possible, to be able to capture new targets. I mean, I shouldn't say target because what we are annotated in the end is genes and proteins, and so well, well, to know if it's a target or not, we will get a clearer picture when we will start annotating the bioactivities, because usually they report on the potential target of the compounds. So hopefully this is coming soon. Regarding the specificity of the disease, yes, I agree. Well, they like to mention dozens and dozens and dozens of diseases in a patent,
Exactly.
and probably at this stage, I'm not even sure if there is a clear ID. There might be there is probably a therapy area. So no, I don't have a solution for that.
Okay, thanks. Could I put in a last request? If you'd increase the frequency of your pushes to PubChem. I'm not sure whether you push or they pull, but okay. Well, I'll give them a jog to pull more often. Yeah.
Okay. Yeah. Please.
Thanks, Nicolas. I enjoy your talk, and I'm really excited that you're adding bioactivity soon. But in the meantime, I wondered, is there a field or a tag that you can use to say, I want to see only patents with data?
You want to see only patents with data? Which data?
The bioactivity data.
Data. Yes, there would be a way of doing that. I mean, the specificity wouldn't be perfect, but obviously we can use regular expressions, or even, I mean, we can use LeadMine even, yes, to do that. Yes, just something to add on the long backlog. But to-do list. Yeah, of course. I mean, we can do that.
Patent biological data often is just a series plus 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 or plus plus rather than actual number. So
Yeah.
how do you capture that?
Well, usually plus plus means greater than 10 micro, and so I mean there is a legend somewhere, and I know that LeadMine is quite good at capturing that. So yeah, okay. It's a range. So we have the right tool for that to explain that.
Okay. I suspect everybody would like to go to the pub, so I think we'll draw it to a conclusion. Please join me in thanking all the speakers. I've been asked to mention that we're going to the Alma today rather than the Pantonomes. So see you all there, I hope. Thanks.
Sounds good. Thanks everyone for joining. Thanks to the speakers. Have a good time at the AMA. See you in a bit. See you next time.